Hello, everyone. Welcome to today's live broadcast, Phase-Appropriate LC-MS Strategy for the Rapid Characterization of Bispecific Antibodies. I'm Lieza, and I'll be your moderator for today's event. We're pleased to bring you this webcast presented by LCGC and sponsored by Bruker with a presentation by LiVeritas BioSciences. LiVeritas BioSciences is dedicated to companies to meet accelerated biologics drug development timelines through rapid and strategic phase-appropriate implementation of mass spectrometry analysis. They offer state-of-the-art LC-MS instrumentation and a broad suite of mass spectrometry solutions. Before we begin today, we have a few housekeeping announcements. First, this webcast is designed to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, and you can find that on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of your slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. And if you have any technical problems viewing or hearing the presentation today, please click on the question mark Help widget in the dock at the bottom of your window. I would now like to introduce today's speakers. We are very pleased to be joined today by Lieza Danan and Parra. Lieza Danan is a serial biotech entrepreneur in the San Francisco Bay Area. Before taking her position as co-founder and CEO of LiVeritas, she held scientific operations and strategic advisory leadership roles in the biopharma industry, including as the co-founder and CEO, COO, CSO at Interven, and as the mass spec function lead to biotech startups, Stemcentrx and Sutro Biopharma. Lieza holds a Ph.D. in biological chemistry from UC Davis and a BS in chemistry from the Ateneo de Manila University. Parra is a senior leader in biotech in the San Francisco Bay Area. Prior to her position as co-founder and EVP of marketing at LiVeritas, Na spent more than 15 years in product marketing and commercialization of scientific instrumentation, with recent roles as the Senior Director of Marketing at Nuomics and as the U.S. Western Regional Manager of Life Science and Mass Spectrometry at Bruker. Now holds a Ph.D. in Analytical Chemistry from UC Berkeley and a BS in Chemistry from Nanjing University. Lieza and Na, thank you for joining us today. Na, please get us started.
Sure. Thank you very much, Laura. Let's get started. So good morning and good afternoon, everyone. Welcome to today's webinar. This is Na, and I want to first say, together with my LiVeritas colleagues and friends at Bruker, we're very excited to have all of you joining us today. Before our CEO Lieza's presentation on the rapid characterization of bispecific antibodies, I would like to take a very brief moment to introduce LiVeritas to you all. The first is, why did we found LiVeritas? So we know that in today's biopharma industry, the industry requires timely and unambiguous data for rapid decision-making in order to advance drug candidates in an accelerated manner. So high-resolution LC-MS analysis offers that sensitivity and specificity to generate the high-quality data needed in guiding drug design and manufacturing processes. Unfortunately, MS turnaround time is often not very ideal to decision makers, which resulted in the exclusion of mass spec as a tool for advancing drugs and optimizing processes. Additionally, we often see insufficient data points and inconsistent data quality, which could also jeopardize making the correct decision. So we, LiVeritas, and our scientists are here dedicated to empower biopharma decision makers like yourself with timely and strategic phase-appropriate mass spec implementation throughout the whole drug design and the development process. So we commit to fast turnaround time, high-quality data to de-risk candidates and overcome process scalability challenges. All right, so how does LiVeritas help? Our solution is called Mass Spec as a Service, in short for MSaaS, which offers a streamlined LC-MS workflow from start to finish. Our capability include project areas as shown here in antibody-derived drug candidates, ADCs, gene and cell therapy, oligonucleotides, and analytical development strategy for your drug candidate that is aligned with your CMC strategy. So MSAT starts with meetings to discuss your project needs and set objectives, followed by submission of samples and the schedule of analysis. So we will make sure to very carefully screen and develop the optimized analytical method for your samples, including sample prep, LC, and mass spec conditions. After data acquisition, we will interpret, inspect the processed data, and present your results and data in the very refined, customized reports that provides all the needed information for your needs. All right, so you must ask, how is LiVeritas different? So here you go. So first thing is convenience. So our clients can conveniently submit their samples at our facility. To many of them, we are very local. Second, we have proprietary software tools to really accelerate the data processing and the interpretation tailored to the scientific needs of our clients. Last but not least is the speed. So we commit to our clients that they can receive the report with actionable insight in as fast as less than a week from the submission, and we guarantee 3 weeks turnaround time. Okay, so our lab is equipped with the ultra high resolution maxis 2 ETP QTOF LCMS from Bruker, which offers that over 80,000 mass resolving power with a sub ppm mass accuracy for very accurate monoisotopic mass measurements of 50 kilodalton, even 50 kilodalton heavy chain, and confident characterization of heterogeneity at a subunit level. The high mass option of the instrument allows for native MS analysis, and the instrument comes with excellent sensitivity in the dynamic range that enables very accurate measurements of drug antibody ratio or values, detection of low-level variants and impurities, as well as sometimes in vivo stability studies of a drug candidate. So finally, the instrument is quite robust, and together with our hands-on expertise, ensures the highly reproducible data will be delivered to you with the precision and accuracy needed for confident identification and quantitation. So to summarize, we are centrally located in South San Francisco Biotech Hub. We're equipped with a state-of-the-art laboratory and LCMS instrumentation, and we offer tailored LCMS service to best address your project needs. But here's our funding team. So our funding team, as you can see, are mass spectrometry experts. And additionally, we're execution-focused industry veterans from biopharma, from mass spec instrumentation companies, academia, and in who we just believe in that mission to support, to help you with drug development, accelerated drug development. So we're passionate about analytical chemistry, mass spec, and the performing science ultimately to help ultimately the well-being of the mankind. All right, to emphasize, this is really one of our key values. We offer packages that emphasize it's the turnaround time. So with the accelerated package, we guarantee the 1 week turnaround time. Even with our basic package, we commit to our customer with 3 weeks of turnaround time. All right, we're thrilled to receive great words and feedback from our recent biotech company clients. Moreover, we're very proud to be the trusted outsourcing partner who offers transformative and strategic MS analysis and solutions for the advancement of their next generation biotherapeutics. All right, so we're proud to share that we have recently formed a strategic partnership with Bruker and have announced the general availability of our South San Francisco laboratory. So we're here, we're ready, and look forward to supporting all of your drug development needs. For more information, you can check our website and download our brochure, contact us, and you'll hear more from Lieza's presentation. Thank you.
Thank you, Nan,
Lieza,
for that. Thank you, Nan, for that introduction to LiVeritas. It is a real pleasure and an honor to be presenting today. Thank you to our collaborators at Bruker and for LCGC for hosting this webinar. We are extremely delighted to have this opportunity to share with you products of our hard work at LiVeritas for the past 10 months since we found our home in South San Francisco. And we have been gifted with biopharma clients with some exciting scientific questions and challenging drug candidates to characterize. So sharing with you in the next 30 minutes or so are some of the stories that we encountered in the past 10 months. Let's start with a general introduction on bispecific antibodies. By design, these are antibodies that engage two epitopes on tumor cells or in the tumor microenvironment in a very simultaneous fashion. Trailblazing advancements on molecular design and genetic engineering have accelerated achieving desirable properties of this group of biologics, which actually led to the development of hundreds of unique bispecific antibody formats out there. There are so many to choose from a design perspective that drug developers will have to choose depending on their desired properties for its intended indication, whether they prioritize the developability of a specific antibody, or they have chosen specific target product features, or specific distribution or pharmacokinetic properties of each of these formats. And then, of course, with so many structural designs to explore, biopharma scientists will have to embrace early the world of what we call process analytics. I was once a part of the process analytics community where we operate in the reality that drug design on paper is not exactly the same as what was produced in terms of batch production in manufacturing, especially when you scale from small scale production to medium scale production to large scale cGMP production. Because the drug produced is composed of different things. These are, of course, the drug product as designed as the major product. And then we have some minor products, which are some minor drug product variants, and including production process-related impurities as part of your minor composition. And collectively, these two minor components are what we call presumptive critical quality attributes. And they have to be well characterized, as some have been known to have negative impact on the efficacy, the toxicity, and the stability of drug candidates. Also, in the world of process analytics and protein analytics, we've come to realize that these minor components, or should we call presumptive critical quality attributes, are actually best characterized and monitored by mass spectrometry-based techniques and assays, especially when providing information on identity, purity, and stability of the drug candidates. And I want to highlight specific activities that has to be done early in the drug design of the candidates. Confirmation of structural integrity is very critical. And of course, later on, towards closer to process development, the identity of impurities and how they are related to the stability of the drug candidates. And all of these data points are important across the board in preclinical drug development, from drug design to IND filing prior to human clinical trials, of course, and beyond that, all the way to FDA approval. In the past, there's what I call the old perspective of limiting mass spec use only for IND filing. And this perspective have resulted to many unwanted late surprises that actually curtailed clinical success of thousands of drug candidates. Luckily, this time around, there is a new enlightened thinking in the biopharma community that focused on the lessons learned in the past decade for drug development, wherein now mass spectrometry has been used in a great expansion from drug design all the way to FDA IND filing and even to BLA filings. And there's a growing interest right now, of course, on how mass spectrometry could be of great service for target discovery. And so this increasing awareness of the power of mass spectrometry for drug development and its utilization demands rapid mass spec data generation while maintaining high quality information. The biopharma decision makers and cross-functional teams cannot wait more than 2 weeks to make decisions to advance top drug candidates for animal testing or to initiate scale-up of manufacturing. This means that fast LC-MS workflows are an absolute need in the entire community nowadays. And so at LiVeritas, what we do is we offer to our biopharma clients a wide variety of fast intact mass analysis solutions with the option of a follow-up peptide map analysis as needed for confirmatory purposes. And this is depending on the phase or stage of the drug candidate. The closer it is to an IND filing, the more we recommend the follow-up testing, follow-up peptide map analysis. But from the beginning stages where speed is of utmost need, we focus on offering these 3 workflows: native conditions or native LC-MS, denatured LC-MS conditions, depending on the needs of our clients in both non-reduced and reduced forms of their samples. And then, of course, we have denaturing LC-MS conditions for subunit mass analysis. That was very helpful for characterization and quantification. All of our analyses are less than 30 minutes, and we try our best to make it even shorter, depending on the needs of our clients. And so in the past 10 months, we have utilized these workflows to characterize over 100 unique bispecific antibodies, and there are of various bispecific antibody formats. And so in the next few slides, I will be sharing 2 particular bispecific antibody case studies, very interesting stories on how rapid characterization have helped our clients achieve their milestones. Even with, in the accelerated drug development world nowadays, you have so many milestones that are need to be met in a very timely manner. And at the end, for case study number 3, I close the presentation with a focus on evaluating the performance of our LC-MS system. So let's start with case study number 1, application of our workflows. for bispecific antibody engineering. We were approached by a biotech startup as one of our clients, a very important client, very exciting scientific questions. The sample presented to us was this bispecific antibody drug candidate. Let's call it Antibody X. It has unknown minor variants that was first detected in process development. The objective of the project was to propose the identity of these unknown variants and perhaps also propose the identity of its modifications. So the findings were to be reported back to drug design or antibody engineering so that they can re-engineer the amino acid sequence of antibody X depending on the findings. And the turnaround time agreed with our client is less than 1 week. The approach we utilize is glycosidase treatment of this antibody sample. We first tested on NIST standard. All of the samples were reduced using DTT and then incubated by 2 different types of glycosidases. Let's call them enzyme 1 and enzyme 2. So here is an example of the results for when NIST was treated with the glycosidase and reduced. Provided here are the monoisotopic masses of the light chain, the average mass of the heavy chain that is deglycosylated by enzyme 1 and enzyme 2. And of course, we compare these results to the glycosylated and reduced heavy chain of NIST. With the same thought process, we applied it on antibody X, incubated it with the 2 enzymes. Here's enzyme 2 plus, here is the reduced heavy chain, incubated with enzyme 2. Overlaid with it is the reduced heavy chain, incubated with enzyme 1. And here is reduced and glycosylated heavy chain of antibody X, showing that enzyme 1 treatment showed removal of up to 4 O-link glycans, with HEXNAC core 1 and core 2 moieties still left intact, and that these O-link glycans have sialic acids remain intact even after enzyme 2 treatment. So when you summarize all of this data together in the table, you can deduce what type of glycans are attached to NIST standard and our antibody X. Apparently, what we found out for our clients is that antibody X has a minor variant that has an O-link glycosylation site. And they were happy to figure out from this, very quick experiments that all they have to do to save this particular drug candidate was to engineer out the O-link glycosylation sites that remained on the heavy chain. The light chain remained unmodified in their production process. So they're very happy about the results. And so the process analytics team members we worked with handed over the information to antibody engineering that modified the amino acid sequence of antibody X. So let's proceed to case study number 2, where we help one of our VIP clients to elect their best drug candidates forward to the next step in the drug development process. So we were approached by a early stage biotech startup with more than 70 unique, submitting more than 70 unique bispecific antibody drug candidates of various formats for rapid characterization. The main objective was for us to determine the bispecificity of each drug candidate Bispecificity is an indicator of its purity and developability. Together with this, they also wanted to identify some of the abundant PTMs or post-translational modifications of the 70 unique bispecific antibodies, trying to screen if any of these are important, presumptive CQAs, critical quality attributes. And so the MS data package that will be generated will be used to back up the election of their top candidates for GLP tox animal studies. The turnaround time for this particular exercise was 2 weeks from submission of samples to preliminary results. There is a huge challenge when working with a large number of samples, and that is the main fact that one platform method for bispecificity will not be applicable to all samples. And the reason behind that is, of course, the fact that we are working with different bispecific antibody formats. And each of these formats has unique structural properties, and they will require different intact mass analysis MS-based workflows. So here on the small table on your left, we've summarized, what we have done. We had to make sure that all of our intact mass workflows are in good shape, that they can address the needs of each of these bispecific antibody formats described on the table. We have different versions of the knobs and holes. And then we also have the first generation bispecific antibody knobs and holes, or what we call the KIH. We've also encountered knobs and holes that are supercharged. And then the most complex version of the bispecific antibodies that we worked with are the multimeric ones, wherein we had to be more creative on how we can derive at most accurate bispecificity numbers. So here we want to highlight how we've utilized native LCMS and how we came to this decision. Here's an example of the raw mass spectra of a supercharged bispecific antibody, the panel at the top, compared to the panel at the bottom, which is a deglycosylated intact NIST antibody. As you can see, the raw mass spectra for the deglycosylated supercharged bispecific antibody is more compressed, and the M over Z range, the M over Z range is lower, much lower than your typical deglycosylated IgG1 sample. The supercharging limits sensitivity and selectivity for what we intended to do, which is to differentiate the major species from the two minor species of this bispecific antibody. So we thought about two different ways of navigating this challenge. One was to remove the acids in the mobile phases in order to minimize supercharging. We also thought about changing polarities, but it's a good thing that the first bullet point worked. What we did was remove the acid, work closer to neutral pH so that we minimize the charging effects and shift the M over Z range to higher M over Z range where the multiply charged species are more properly spaced apart. And so as a result of this, we were able to calculate bispecificity of our supercharged bispecific antibody samples. And here's an example of the deconvoluted mass spectra showing the different percentages of the major species and the two minor species. And so with native LCMS being able to solve this issue, we went on and started analyzing over 70 bispecific antibodies, determining their bispecificities. And here they are, 16 samples of knobs and hole format and the multimeric format showing the main species of species 1 and the 2 minor species, and also for the multimeric forms of samples we received, looking as well for the bispecificity of species number 2. And then the rest of the samples of over 50 samples were analyzed using native LCMS. Again, most of them are supercharged molecules, and so they were very helpful. Native LCMS is very helpful in determining bispecificity, again, differentiating the main species from the minor species of this bispecific antibody samples. And of course, some of them doesn't necessarily have to work in native LCMS, so we switch back and analyze them in denaturing LCMS conditions. And so with that data, our clients were able to advance their top 3 candidates to the next step, which was the GLP tox animal studies. Our final case study focuses on evaluating our LCMS workflow performance. As Na mentioned earlier, what we have at LiVeritas is the Bruker maxis 2 ultra high-res QTOF mass spectrometer coupled to an LC system. So when we are analyzing large batches of samples, what I typically do as part of our sequence is add different system stability metrics to evaluate LCMS system performance across many days. This becomes our basis to pass or fail the newly acquired data set. So our objective for this case study is to evaluate the LCMS system performance for relative quantification and large batch analysis, intact mass analysis, intact mass samples. Our standard is the NIST monoclonal antibody, and our sample is a highly glycosylated antibody that's called antibody Y or AbY. So for preparations, we've prepared both the standard and sample in its non-reduced deglycosylated form and also in the reduced deglycosylated form. These are some of the metrics that we have tested for precision. We looked at both for mass accuracy and for integrating peak areas or intensities. We looked at method precision, intraday precision, and intermediate precision. And we also typically look for detection limit and linearity. For this particular exercise, we utilize BsAb from 5% to 95% in terms of composition relative to another antibody in the sample, antibody Y. And for specificity, we looked at the mixture of 95% antibody Y versus 5% NIST antibody standard. Antibody Y is a highly glycosylated sample. Here's a deconvoluted mass spectra of antibody Y that shows you the mass of deglycosylated antibody Y around 139 kilodalton, and still various up to 4 or 5 glycoforms still remaining intact in the sample, which are indicative, which indicates incomplete deglycosylation. So, as we know, the BsAb standard, when incubated with glycosidases, are completely removed. The glycoforms are completely removed. So we're working with 2 samples where one we expect to be more ionized compared to antibody Y, which we are expecting to have lower response factors. Here is spectra, the convoluted mass spectra showing the mass accuracy of intact NIST antibody standard that was maintained at less than 12 ppm after 85 injections analyzed in about 4 days. So injection number 1, at the start of the sequence, we have mass accuracy of 4.5 parts per million. In the middle of the sequence, it was injected again at 5.7 parts per million. And after another 25 injections, we injected NISMAB again. The mass accuracy remained at 5.4 parts per million. And then at injection number 85, which is the end of the sequence, there was a deviation, a slightly higher deviation in mass accuracy. We noticed at that particular day, the temperature inside the lab changed and became warmer, which actually affected our mass accuracy and went up to 11.4 parts per million. I want to take note that we've only performed external calibration for this particular exercise. So after this exercise, we started employing internal calibration to ensure that our mass accuracies are all below 10 parts per million throughout our long batch analysis. And so here what we wanted to show is, again, the average mass of deglycosylated NIST intact monoclonal antibody. We can also detect, as you can see very clearly, plus 162, which is NISMAB plus a glycation or plus glucose. For this particular number of injections, n is equal to 27, showing with you that the coefficient of variation is definitely is much lower than 1%. We've also performed the same exercise for the reduced and deglycosylated NISMAB, looking at the mass measurements at for the light chain, the monoisotopic mass, and then also the average masses of reduced glycosylated heavy chain, monitoring 3 different glycoforms. So for all of these 4 mass measurements, the coefficient of variation of percent RSD are much lower than 1%. They are in the third and fourth. They're in the 3rd decimal place, which gives us so much confidence in the system that we're using for intact mass analysis, regardless of the mass range we're looking at, from 20 kilodalton all the way up to 150 kilodalton. Here in this slide, we're presenting to you the linearity of NISMAB, non-reduced and deglycosylated. We have evaluated concentration from 5% to 95% NISMAB. It's clearly shown here that even with using total ion count chromatogram for the peak areas, we have a linear calibration curve. And based off that linear calibration curve, our detection limit is at 13%. This is meant for quantitation purposes. For qualitative analysis, of course, our detection limits are much lower than 13%. We can detect intact NISMAB at 5%. We've also done the same linearity exercise for the NISMAB reduced heavy chain. Showing here is the full calibration curve from 5% to 95% based on this calibration curve with an R-squared of 0.9680. The detection limit is at 10%. However, if you look at the residual plot to your right, it shows a downward V-shaped pattern, which indicates that a simple linear regression model may not be the suitable calibration curve for the deglycosylated heavy chain using an XIC of MRZ 934.3. We've also evaluated a non-linear regression calibration curve, and that provided us with an R-squared of 0.99. So we are always comparing, and open to looking at different ways to have the most suitable calibration curve for our intended purpose. We also evaluated the detection limit and linearity of the heavily glycosylated antibody Y. Here we're presenting reduced and deglycosylated antibody Y heavy chain. It is showing here that the very low response factor for 5% antibody Y is consistent with the detection limit of 10% from the previous slide, specifically for heavy chains. And so what we can do for antibody Y is be able to quantify it confidently from 10% to 75% composition as part of in our calibration curve. And last but not least, I just want to show you what I promised earlier, that we know for a fact that qualitatively we can detect deglycosylated BsAb at 5% intact antibody, which gives us good indication that our sensitivities could even be much lower than 5%. So if you look at the intensity right here, it's about 1,000 units. If you bring down, had we brought down our sample to less than 5%, I'm sure that we can still see deglycosylated BsAb around 500 units in terms of intensity. And here's the corresponding deconvoluted mass spectra, showing the average masses of your deglycosylated intact BsAb. And again, our heavily glycosylated antibody Y. At 5%, at 95% deglycosylated antibody Y, we can definitely still see here the 5% BsAb antibody. So I hope you find our stories, our case studies interesting and useful. We're happy to share with you this information. And I hope you connect with us if you have any mass spec analysis needs. Please take a look at our website at LiVeritas.bio. I want to use this opportunity as well to thank our collaborators at Bruker, Yum Trementine, Michael Gregg, Liling Fang, Chris Swift, Lauren Upton, Paul Spear, and Ron Thakur, of course. And of course, I want to thank the LiVeritas team, specifically our more recent add, Chris Dancel, to the team. And to the audience, thank you so much, of course, for your time and for your questions. Happy to answer them shortly. And thank you, LCGC. Handing it over back to you, Laura.
Thank you, Lieza. Thanks for that excellent presentation. It is now indeed time for questions from the audience. As a reminder to all of you in the audience, if you would like to submit a question, you can do that by typing it in the Q&A box, and that's on the right-hand side of your presentation window. We've had a few questions coming in already, so let's get started with those. Here's one to start with. Is the site of O-glycosylation identified with MS data?
Not for this particular exercise. We leave it up to our clients to make that decision, whether they want further confirmatory or not. they have not requested for that particular analysis yet.
Okay. Here's another question. In slide 28, which mass spec did you use to get MS data from the bispecific antibody? And if you want to push that slide out, you can. If you hover over it, you can hit push to audience.
So all of the analysis for this presentation, we have utilized the Bruker maxis 2 ultra high res QTOF for intact mass analysis.
Excellent. Another question in slide 28, did you see heterogeneous species of antibody?
Let me see. How do we advance all the way to slide 28?
So you can actually, maybe I can even do it for you. You can go to slide 28, hover over it, and hit push to audience. So, yeah, I think I'm seeing that on the screen, if that's the right one. Supercharged bispecific antibody.
Okay. Can you repeat the question, please?
Yes. It says, did you see heterogeneous species of antibody?
For this particular exercise, because of the loss of sensitivity and selectivity, we did not see it. But the moment we switch to native LCMS, yes, we did see. We did see it in native LCMS.
Excellent, thank you. How much sample cleanup is required before working with heterogeneous proteins like bispecific antibodies?
Not much. For this particular exercise, we have employed an LC workflow that allows us to remove any excess buffer early on. So it's not much cleanup in the front end. And our goal with doing this is to make sure our execution is rapid. So any sample prep will be on column. On any desalting that we do is on column. Sorry, not any sample prep. So we have our sample prep methodologies, of course, but then any on column, any desalting needed that may interfere with the mass spec analysis will be performed on column.
Okay. Another question. The NIST map spectra show a leading edge for each glycoform. The deglycosylated spectra look good. Any ideas about what's going on here?
Oh, hi, Vish. Maybe we can take that offline and answer that because I need to look at the spectra and a bunch of other information so I can answer this more clearly and more elaborately. I'm happy to connect with you, Vish, to answer your question.
Okay, great. Here's another question. For native LCMS, what type of column did you use?
Oh, it's a column from poly-LC that was first used by Seattle Genetics back in the day. Let me try to remember it. It's called the polyhydroxyethyl column. Andrew Alpert will be very happy to help you. This is the same column that I've used in the past for antibody drug conjugates, drug load distribution determination. It works nicely for native LCMS. Unfortunately, though, I can share with you that for some of our multimeric samples, they crashed on column. So it's possible Andrew Alpert may have developed a brand new poly-LC column for this type of more hydrophobic samples that are not stable at neutral pH.
Great. Sorry, I'm muting myself. For native LCMS, have you used ion exchange as your separation chemistry?
Not at the moment, but that is definitely something to be explored.
Another question going back to slide 28, was the pH crucial for the native mass spec? And what did you use to substitute the acid? Some mobile phase modifiers?
Yeah, so it's a typical, again, going back to the methods. I wasn't able to publish this method in the past, but I think folks at Cell Genetics was able to publish this back in 2018. There's a MABS journal article that describes this. It's a typical ammonium acetate buffer. You don't even have to adjust the pH. Some folks would prefer to adjust the pH to 7.0. You can keep it at 6.8.
Excellent. Sorry, I had a question here. How much tuning is needed to optimize from peptide mapping to protein analysis?
Well, we're very lucky to have some pre-tuned tune files already saved in our system. So not much. There are specific parameters. I would say there are at least 5 parameters you have to look out for in the maxis to switch for low molecular weight detection to high molecular weight detection. And you just keep on monitoring these 5 specific parameters and operate in, predetermined ranges ahead of time. Then you'll be able to switch back and forth quickly. It's common in our lab to be performing, intact mass analysis early in the week, and then the latter half of the week could be peptide map. And the tune files are very sturdy, and as long as you keep your source clean, there shouldn't be that much problem for tuning.
Can you specify from your performance metrics your method precision limits, percent of expected, et cetera?
Right. For method precision, what we wanted, so best practice from back in the day, myself working in the lab, my method precision for relative quantification always has to have a coefficient variation less than 20%. If I'm actually close to 20%, it's not good. I actually want to set a bar higher, less than 10%. For mass accuracy, of course, it's expected that the coefficient of variation to be less than 1%. Cannot be more than 1%. So I'm very happy with the performance of the maxis for our intended use. The coefficient of variation for, how many days we're using it could be much less than our set standard of 1%, less than 1% coefficient of variation. So.
Did you encounter any challenges while characterizing O-glycopeptides with ETD as MsMs?
Can you say that again, please?
Yes, I'm not sure if I read it right. It says, did you encounter any challenges while characterizing O-glycopeptides with ETD as MsMs?
So as I said, we haven't done ETD MsMs for this specific samples. Not yet. That will be the next webinar.
Fantastic. Okay, just checking to see if I've got some more questions here. Just one other question. How does LiVeritas manage data security for biopharma clients?
Oh, sure. Happy to answer that. Well, what we do is we're very serious about data security. And so what we do is we anonymize and encrypt client-related information in our systems. And also we have actually tiered and password-protected access to all of the LiVeritas data and analytical systems.
Excellent. Another technical question. How could you solve the solubility for some highly hydrophobic MABs? Did you perform a solubility assay What about the reconstitution solvent before LCMS?
Oh, well, most of our clients keep their samples in PBS. And with PBS, we didn't experience any solubility problems. So it's now solubility, when you switch buffers from PBS to ammonium acetate, is not a guarantee. So that's when you have to be creative in how to handle the situation. Well, like you start asking yourself, is this meant for native LCMS analysis, or should we employ a totally different strategy to analyze this particular sample for bispecificity? Maybe we should just switch back to denaturing LCMS, or maybe you need to do some enzymatic incubation to slightly improve the hydrophobicity of this hydrophobic antibodies.
Very good, thank you. How does LiVeritas ensure timely delivery of high-quality results?
Well, one thing I learned from working in the lab for like at least 7 years in the past, the speed of mass spec analysis is dependent on the tools that you have in front of you. And so what we have at LiVeritas is a unique set of in-house developed proprietary tools that allows us to very rapidly churn data. Of course, we utilize vendor-supplied software tools as well, and then we complement that with our proprietary tools to ensure that within our promised delivery times, we will be able to provide you with the results that you need.
Fantastic. Well, with that, I think we should wrap up. Thank you so much, Lieza, for that excellent talk today, and to Na as well.
Thank you so much, Laura, and thanks everyone again for attending our webinar.
Excellent. And I'd also like to thank our sponsor, Bruker, for making today's webcast possible. We'd like to ask all of you in the audience to participate in a brief survey. You can see that survey on the right-hand side of your screen. You'll receive an email alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.